Right, so I'm going to pick up from where I left off last time. So we saw last time the definitions on a couple of examples uh, of synthetic media, defined media, and complex media. Now, attends, mais c'est encore ça. Okay. So as well as basic, basic complex media with just 2 or 3 ingredients, there are also what are known as enriched media, and these are complex media to which something has been added that is a nutritional requirement for a particular type of bacterium. So one which is used rather frequently is fresh blood agar, which has got casein, tryptones, and peptone digest of soya, some salt, and it's supplemented with 5% sheep's blood. So you get these red agar plates on which some bacteria can grow. So they're very useful for growing up what's called fastidious chemoheterotrophs. So these fastidious bacteria won't really grow very well on a medium like LB agar. So there's something inside these eukaryotic cells in the blood that is a nutritional requirement for these bacteria. So streptococcus and the listeria don't grow very well on LB agar. They grow pretty well on sheep's blood agar. So for example, in the TP classes, in the lab classes, you will be isolating environmental bacteria on LB agar plates. So probably you won't be able to get any streptococcus growing on there because they don't grow very well on LB agar. Even though, you know, there might be some in the sample. Now, the next type of medium, which is, you know, extremely useful for a lot of purposes, are selective media. So selective media contain some kind of components that inhibit the growth of certain bacteria or promote the growth of the bacteria that you want to isolate from a kind of complex mixture. So any kind of sample like comes from the ground or comes from feces or something like that, there are going to be a lot of bacteria in there. You might just want to find out, do I have the presence of particular type of bacteria? So in this case, you might want to use a selective medium. So for example, Chapman's agar has or contains 75 grams per liter of sodium chloride. So only halophiles or halotolerant bacteria can grow on this medium. So most bacteria will not grow, but halophiles and halotolerant bacteria like staphylococcus can grow. Another selective medium for staphylococcus is Baird-Parker medium. So this contains lithium chloride and tellurite, which inhibit the growth of most gram-positives and gram-negatives. And it also contains as a carbon source pyruvate and glycine, which are used preferentially by staphylococcus. So they can grow very well on this medium. And most other bacteria, even if they can survive the tellurite and the lithium chloride, won't grow very well on these carbon sources. And the final example here is McConkey agar, which has got bile salts and crystal violet, and these inhibit the growth of most gram-positive bacteria. So this medium is going to be selective for gram-negatives. Now, a lot of selective media are also differential, and that means they contain some other component which will cause a color change for some particular types of bacteria. Okay, so in Chapman agar, the differential component is the presence of mannitol. So mannitol-positive bacteria that can ferment mannitol will reduce the pH of the agar and cause a color change. Now in Baird-Parker, the tellurite has also got a kind of differential quality because bacteria that oxidize tellurite to telluride will cause a black deposit. So you get black colonies. And the other thing that is differential in Baird-Parker is the presence of egg yolk. So this means that the agar has got a translucid quality, and which will become clear if bacteria are secreting a lecithinase enzyme. So that means that black colonies with a clear halo, that's definitely Staphylococcus aureus. And McConkey agar, the differential component is the lactose. So once again, bacteria that can ferment lactose will cause a color change. And gram-negative lactose positive bacteria are generally known, generally coliforms. So this is kind of what you can see. So on a Chapman agar, there's two Staphylococcus species that can grow here. One of them is Staphylococcus epidermidis, so it resists salt, it can grow, but it doesn't ferment mannitol. The pH of the agar stays neutral, and the neutral red indicator that is there, or the phenol red rather indicator, stays red. Whereas here, where you've got Staphylococcus aureus, this bacterium is mannitol plus, ferments mannitol, the pH of the agar goes down, and the color changes to yellow. Baird-Parker rather, sorry. You get these black colonies with a clear halo, definitely Staphylococcus aureus. And here on McConkey agar rather, you have these very bright pink colonies. These are lactose positive coliforms, whereas this one here, this is a clear colony, lactose negative. It's gram negative and lactose negative. Okay, so the useful thing about these selective and differential media is that they allow you to isolate and identify bacteria in a single step. You get the sample, spread it on some differential media, and you say, yeah, okay, I've got Staphylococcus aureus in my sample. I know it, it's there. You get the result as soon as they grow. Okay, I don't know if I've actually renewed the link on this, so let me just try and copy that. Might not be correct. See if everybody has understood the question here. Okay. Yeah, that should be good. Just to see if everybody has understood the concept here. Okay, let's see what we got. Okay, so we've basically got kind of three questions. Define complex, selective or differential for salmonella, and selective or differential for gram negatives. Okay. So the first one, define medium or complex medium. It's a complex medium. Why is it a complex medium? Okay. Can anybody tell me the list of the exact chemical components of a meat extract? No, nobody can tell me that, right? You don't know precisely what is present in this culture medium. You don't know all the chemical components that are here. That's why it's a complex medium, because it has this kind of like complex mixture, peptones and meat extract. So complex medium. Defined medium, you have, glucose, salts, you might have one specific protein at a particular concentration, this kind of thing. But that's not the case here. So it's complex medium. Now the next thing is selective or differential for gram negatives. Now, I think this is an important concept here. So most people put differential. And what does that mean? What that means, if it's differential, is gram positives and gram negatives can grow on this medium, but the gram negatives will be a different color so you can identify them. That's what differential medium is. So are gram negatives going to give you a specific something on this medium? No. Is it selective for gram negatives? And in order to answer that question, you have to find out whether there's something in the composition which will inhibit the growth of gram positives. And there is something. Can anybody see what it is? Okay, so we had an example of a selective medium for gram negatives just 2 minutes ago. That's the McConkey agar. Yeah. Biosalt, yes. This is the one. This is the thing that's in common between these two. So it's going to, so the biosalts here will inhibit the growth of gram positives. So that means it's selective for gram negatives, not differential. Okay. And the last one is differential or selective for salmonella. So it's the same kind of question. If it's selective, that means only salmonella will grow on this, okay? If it's differential, salmonella can grow and something else can grow, but the salmonella will be a different color. And that's correct, isn't it? Because it says Shigella give you clear colonies and salmonella black colonies. So in fact, it's differential for salmonella. So this is complex medium, selective for gram negatives, and differential for salmonella. Salmonella. Yeah, that's right. And it's also got lactose, so coliforms like lactose positive, gram negatives, they'll give you maybe pink colonies. So you have pink, clear, and black, and it will give you something differential between Shigella, salmonella, and coliforms. Okay, so selective and differential media, very useful. Yes? Kind of not really, because it's mostly kind of by elimination. That is, There's nothing specific about the Shigella colonies here. They just they don't have a specific color. Or you can say, okay, so no, another thing, right? We're going to see this in the lab class a little bit. So, and okay, so why are these colonies black anyway? Okay, so what is happening is here is that the Salmonella can use thiosulfate as an electron acceptor and reduce it to sulfide, and it precipitates out with the ion. Okay, so that means basically in biochemical terms, we say Salmonella are H2S positive. Okay, they can produce sulfide. They have this biochemical phenotype. The lactose is here to find out if bacteria are lactose positive. They can ferment lactose. So basically, what you have here is you have gram-negative bacteria that are lactose negative, sulfide positive, and in really most cases, these are Salmonella. Okay, but for the Shigella, the information you have is lactose negative, H2S negative. And Shigella is one of the bacteria that has that phenotype, okay? But there are others. So it's not very specific because you've got like negative information. It's always, always bad to give a positive identification just because something is got lacks this, lacks that, because there'll always be some other bacteria that have this, that share that negative phenotype. But good question, though. Thank you. Okay, so what we need to do for the next hour is think about bacterial growth, how we measure bacterial growth, how we measure the rate of bacterial growth, and then how different environmental factors can have an effect on bacterial growth. Now, in order to do any of this, you have to be able to quantify how many bacteria you have in a suspension. And perhaps really, maybe not the simplest, but the simplest or the most direct way to quantify live bacteria in a suspension is by, well, making a serious dilution of your starting sample. So here you've got 1 ml pipetted into 9 ml of water, 10-fold dilution, and 1 ml of that comes out, so you get serial 10-fold dilutions. And then in this example, what they do is they mix 1 ml of each of these dilutions together with 9 ml of agar and pour it out. Put it in the incubator, and then here you have 50 colonies that have grown up. Here you've got 5 colonies. And for the calculation, what you do is you choose the colonies with between 30 and 300 colonies, the agar plates rather. Some people say 25 to 250 colonies. So plates with more than 300 colonies, you can't really count them accurately because you have bacteria that are growing one on top of the other, and you can't really be sure that one colony is really one bacterium. Whereas plates with less than 25 colonies, statistically, is not very reliable. If you did a whole series of them, sometimes you could get 10, sometimes you could get 15. There's a lot of variability in the measurement. So that's why people say, okay, something between 25 to 250, that's the best. So here, you use the plate with 50 colonies. And then what you do is you multiply the number of colonies you have by the dilution factor, and then you divide by the volume that you spread on the plate. So here we have 50 colonies at the dilution 1 in 1,000. So the dilution factor is 10 to the 3. And the 1 ml was used to spread the plate. So that gives you 5 times 10 to the 4 colony forming units, or CFU, per ml. Generally speaking, 1 CFU is 1 live bacterium, because when they were spread out, before you put them in the incubator, you've just got 1 bacterium here, which you can't see because it's too small. But as they grow up, they form a colony. of hundreds and thousands of bacterial cells, and you can see them. So that's the classical way of counting live bacteria in a suspension. Now, the other thing you can do is make use of the fact that when bacteria grow, then the medium becomes cloudy. Okay, so this is a tube cultured, no bacteria, it stays clear. Bacteria in there, it becomes cloudy. And the turbidity of a suspension can be measured by turbidimetry on a spectrophotometer. And the basic idea is if you transmit light with a wavelength of 600 nanometers through a solution with no particles suspended in it, then all the light gets transmitted. Whereas if you pass the light through a suspension of bacteria, some of the light will be deviated, deflected, so you'll have a reduction in the transmitted light. Okay? And this will be measured by the spectrophotometer as an optical density. So if you measure the OD, the optical density, at 600 nanometers, there's a linear relationship between that OD 600 and the number of bacteria that are in the suspension. And that's just an application of the Beer-Lambert law. So that means what? That means that, you know, your OD 600 is equal to the number of bacteria multiplied by some kind of constant. So in biochemistry, if you've got some kind of compound that absorbs light at a particular wavelength, what do you call this constant? Have you done this yet in TP? Spectrophotometry, you have some kind of solution and you measure how much the light is absorbed at a particular wavelength, it tells you the concentration. Yes, yeah, the extinction coefficient, okay? So for a compounded solution, it would be the extinction coefficient. Here, we've got particles in suspension, so it's not the same, it's not really an extinction coefficient, okay? So for bacteria, basically one OD 600 unit is equivalent to 10 to the 9 bacteria per ml. And that coefficient is variable from one bacterium to another. It's not exactly the same for every species of bacteria because bacteria are different size, they're different shapes, so different bacterial cells will deflect light more or less well, okay? However, once you measure this extinction, well, the relationship between OD 600 and the number of bacteria for a particular species, then that constant, you can use it all the time, okay? So what does that mean? You know, the exact ratio different for different bacteria. The other thing you have to remember is that you can't tell the difference between live and dead bacteria with turbidimetry. So it's a total live plus dead. Another thing perhaps to notice about this technique compared to the plate counts, which one is going to be the most sensitive technique, which can detect lower amounts of bacteria? This one or what we just saw? Yes, colony counting is going to be more sensitive. Why?
Because I don't know if this technique is very precise because you can't really know how many bacteria there when you see the lights reflected.
Well, you can know, but it will only be precise within a particular range of where you can quantify it. So if you've got an OD 600 measurement 0.01, is that going to be an accurate, a valid measurement with the sensitivity of the spectrophotometer? Not really. I trust the values that are 0.05 and greater. 0.01 is too low. And that's already 10 to the 7 bacteria per ml. So basically 10 to the 7 or 1 million bacteria per ml, you can't really detect them accurately here. Whereas you can detect them and count them accurately with the plate count, okay? So 5 times 10 to the 4 per ml, you can detect them here. But 5 times 10 to the 4, that same suspension here, it will be completely clear. You won't be able to detect with the OD600. Okay, so there's a question about the limit of detection, which is different. And that's because of this. 1 OD600 is about 10 to the 9 bacteria per ml. Okay, so that's how you can measure how many total bacteria and how many live bacteria I have per ml in a liquid culture. And once you can measure how many bacteria you have and count them, then you can look at the characteristics of their growth in a closed system. So in a closed system, for example, I mean you have maybe 200 ml of LB medium, and you add in 5 ml or 2 ml of a starter culture of bacteria, and you shake it at 37 degrees, and you take samples at different time points, and you measure how many bacteria per ml you have using the agar plate technique. So you're only looking for live bacteria. And I know this because on my scale here, I've got the units of measurement. This is CFU per ml, so it's done with the plate count. And what is always important is when you do something like this is to use a log scale. We'll see why this is important. So when you do this experiment, what you can see is bacteria will grow in 4 phases. Now the first one is the latency or the lag phase. That is bacteria that you've kept, the pre-culture you've kept in your fridge overnight. When you put them into fresh medium at 37 degrees, the bacteria you inoculate are not yet ready to grow. You know, they've been kept in a fridge somewhere for a couple of days. They've shut down their metabolism. They've gone into a kind of like survival phase. So once they get inoculated into a rich medium with nutrients that are available, the temperature is optimal, then they have to reactivate their metabolism, start expressing autolysins to break down the peptidoglycans, start expressing glucose transport molecules for importing the glucose, the enzymes involved in glycolysis and generating metabolic energy. So this takes time, okay? So you can have a 20 or 30-minute lag phase. However, once they start to grow, then they will grow exponentially. And I know that this is exponential because we've got a straight line on a log scale. So as long as you've got a straight line here on a log scale, then you're in the exponential phase. Now, after a while, bacteria will stop growing. That's because either they've used up all the nutrients in the medium, or there's a buildup of too many toxic compounds that are the products of their metabolism, and that will also stop growth. So typically for bacteria that grow using fermentation, they will excrete acidic fermentation products, so the pH of the medium will go down, and this can inhibit their growth. So you'll have a stationary period for a certain amount of time. They've just stopped growing, and here they're switching into the survival phase. Okay, so they express fewer autolysins, they stop peptidoglycan synthesis, they'll shut down a lot of their metabolism. So they're waiting for things to get better, but if things don't get better, then they go into the death phase. You'll get fewer and fewer viable bacteria at these later time points. Now, sometimes people talk about the 5 steps, 5 phases in a bacterial growth curve, because if you extend this experiment even longer, what will often happen is the bacteria, they don't go down to 0. You'll have maybe 1% or less of the initial population, which will survive for a much longer time. And Well, there are different explanations for that, which I don't really want to go into now. But sometimes you see in textbooks 5 phases of bacterial growth, and the 5th one is like the long-term survival. Yes.
What caused this?
Hmm?
What caused this?
Well, they're running out of nutrients. They can't, they don't even, because even if they slow down their metabolism, they still need something. So they can't like live totally with nothing. They're not like bacterial endospores. They're metabolically silent, nothing, no activity. So they can survive for years, basically. But a vegetative cell is still got some kind of basic metabolic processes. And if they come down, what can happen when proteins denature in a bacterial cell? There's no energy left to like resynthesize them. What happens if DNA gets damaged? There's no energy left to like repair it. So then these things accumulate. The bacterial cell might still be there. It's not like exploded, but it won't be able to grow anymore because it's just accumulated so much damage. It's not viable. So when you like spread them on the agar plate, it won't form a colony. Okay, so that's the different phases of bacterial growth. And this exponential phase, why is it exponential? It's simply because bacteria grow by binary division, right? So when you start, one bacterial cell will divide into 2. And then in the next generation, these 2 cells will divide into 4, and then 8, and so on. So you have a very direct mathematical relationship between the number of generations and the number of bacteria that you have. So it's basically just these powers of 2, okay? So in generation 0, you have 2 to the power of 0. First generation, 2 to the power of 1, 2 squared, and so on. Now that is helpful because we can use it to calculate some, numbers which will describe how fast bacteria are growing. So, and one of the things we want to calculate is what's called the generation time. How long does it take for a bacterium to divide into 2 cells on average? And the way to calculate the generation time is this. So if we've got this relationship between the number of bacteria at the time t and the number of initial bacteria, that's the number of initial bacteria times 2 to the power n, where n is the number of generations of bacterial division we've had. Now, what we can do is we can measure these 2 things, bacteria at time 0 and bacteria at time t. So when I say time 0, that means the start of exponential growth. So on the graph here, time 0 for this equation doesn't really mean time 0 here. It means this when exponential growth starts. So it's time 60 minutes. That will be our t0 here. And then you've got a kind of pretty much linear relationship here. So any one of these points you can use. So you could have time 60 and time 160. Okay? And we know the number of bacteria that are present at these time points. That's what you read off on the y-axis here. So we know this number and we know this number. We just want to find out little n over here. And in order to do this, we will use the magical power of logarithms, because if you take logarithms on this kind of equation, first you get something like this, and then that allows you to bring the little n down over to the front here. And once you've done that, then you just rearrange it and it gives you this equation. So we know this, we know that. This is just some kind of number, you can look it up. And that means we can calculate the number of generations that have occurred. And since we also know the lapse of time, so if we're using time 60 for our time 0 and time 160, that's 100 minutes, then we can calculate the generation time. So that's the time that's elapsed divided by the number of generations. And this is the average time required for a bacterium to divide. So the faster the bacteria are growing, the shorter this time is. The slower the bacteria are growing, the longer this time is. So just some before I do that, yeah, I should maybe rearrange the slides here because I'm really getting onto this. So I'll rearrange these slides, just put this one up here instead. Yeah, so here are some examples of generation times for different bacteria. So the kind of things that grow pretty well in culture, like Escherichia coli, Staphylococcus aureus, they will have generation times of less than 1 hour. So this is something like 20 minutes. This is something like 30 minutes. Under optimal growth conditions with optimal medium, they can grow that fast. So that means you put the culture in overnight, the next day you've got a lot of bacteria, or you can see colonies on a plate. They're easy to work with. Other bacteria, like Mycobacterium tuberculosis or Treponema pallidum, which causes syphilis, their growth or generation times are much longer. So if you want to grow Mycobacterium tuberculosis colonies, you can do it. There are selective media for that. But you have to put them in the incubator and you come back in 3 or 4 weeks. It takes much longer for colonies to grow. So some important pathogens only grow slowly. And another thing that is also true is that autotrophs tend to grow more slowly. So Anabina cylindrica, this is a cyanobacterium, photolithoautotroph, Nitrosomonas, chemolithoautotroph. Their generation times are longer, even in optimal growth conditions. And you can kind of understand it because they need to perform a lot more biochemical work. These guys are living on carbon dioxide. So first they've got to reduce the carbon dioxide and then they've got to build it up into more complex organic molecules. It takes time to do that, okay? Whereas for E. coli, Staphylococcus aureus, they're chemoheterotrophs. If you give them a rich medium, they're importing glucose, peptides. They don't have so much work to do, so they can grow faster, okay? So just some examples of generation times. Now, one other thing that everybody needs to understand about doing this kind of calculation is the importance of the log scale. And so here's a set of results here that you might get. And if I plot these out on a linear scale, then it looks like this is the kind of maximum phase of growth for my bacteria. It looks pretty linear along here. And so I might say, okay, my time 0 is 100 minutes, and then I'll use the time 180 minutes over here. Now, if I plot the same data on a log scale, I can see that this is rather misleading, okay? Because what this is telling me is that my exponential growth phase starts really here at 60 minutes, and it goes up to maybe 120 minutes, or maybe 140, but basically even here it's slowing down. So 60 to 120 minutes. So if I plot my results on a linear scale, it's going to give me a false impression, and then I'll use the wrong time points for the calculation, and the result will be wrong. Whereas if I use a log scale, that's what allows me to see when my exponential phase begins and when it's starting to slow down. And then I can get the right number from my calculation. So everybody should know this when you're getting your results in the TP. Okay, so what have we seen so far? Well, from last week, nutritional requirements and nutritional types, not really the same thing. Auxotroph, that's to do with nutritional requirements, will require some additional growth factors. Autotroph can fix CO2. Import mechanisms for different types of molecules. ABC, ATP binding cassette, the most expensive, then proton symport mechanism, slightly cheaper, group transport, most economic way of getting glucose molecule into the cell. Siderophores, essential for iron and other metal ions at lower concentrations. And then what we've seen different types of medium, and the bacterial growth curve in liquid medium. Okay, so I don't know why that's there. That's kind of weird that I have that there as well. It's in the wrong place. So sorry about that. So yeah, should have just switched over that really. So the other thing that we should all be aware of is that bacterial growth in liquid medium is only kind of half the story, because a lot of bacteria grow on surfaces, either alone or in consortia. So single species, and these, when bacteria grow on a surface, they secrete some kind of polymer around them and they grow in it. So this is what's called a biofilm. Got bacteria and some kind of polymer that they secrete. So there are some single species biofilms. So Pseudomonas aeruginosa secretes a very nasty biofilm during persistent lung infections, makes it very difficult to get rid of them. Staphylococcus aureus, same kind of thing on medical implants. Now, a lot of bacteria grow in multi-species biofilms. So they are not growing alone. There's a kind of whole microbial community that's growing together in the same film. So dental plaque, at the base of your teeth, the dentist goes and scrapes out periodically. This is a very, very interesting bacterial consortium, and the plaque is produced by the bacteria that live there, and you have different types of bacteria that live at different levels in the dental plaque. Okay, so a lot of bacteria grow in biofilms, and growth on biofilms follows a cycle as well. So initially, what will happen is that planktonic bacteria that are floating around in a liquid medium will attach to a surface and become sessile or immobile, attached to the surface. And this attachment is initially quite weak, so they can be washed off easily. But then they will attach definitively to the surface. And this is by the expression of particular adhesins, which are often bacterial pili. So they'll adhere strongly to the surface. And then they'll start to grow on the surface. Now, at a particular point, when the bacteria are sufficiently numerous, they will all start to secrete an exopolymer. And that's really when the biofilm starts to take on its kind of gunky, slimy character. So after the synthesis of the exopolymer, then this whole thing will start to grow. So the biofilm will mature, and then finally it's going to grow too big. The bacteria on the inside of the biofilm won't have access to nutrients anymore. So there'll be a dispersal phase. Once again, this is a concerted behavior by the bacteria in the biofilm. They'll start to secrete enzymes that degrade the exopolymer. They'll start to express flagella, which will allow them to swim away and try and go and colonize another surface. So we have this controlled cycle of growth in biofilms, with bacteria switching between planktonic and biofilm phases. Now, one of the interesting things about this is the kind of what's called, concerted behavior, collective behavior of the bacteria growing in a biofilm. And this kind of collective behavior was first detected in Vibrio fischeri, which is now officially called Alii Vibrio fischeri. So these are gram-negative bacteria that live in symbiosis in the light-generating organs of some particular species of squid. Okay, so these squid, they've got spots on their skin that produce light. And it's the bacteria that live in those spots that are making the light. Okay? And in order for this light to be produced, the bacteria express an enzyme called luciferase. And in order to produce the light, all the bacteria in the organ, they've got to produce the luciferase at the same time. So the people who were working on this at the end of the 1970s, they isolated the bacteria from the squid, and they started to grow them to see, how they turn on and off the expression of this luciferase enzyme. And what they discovered is that the thing that causes them to all start expressing luciferase and produce light is when they're growing at a very high density. There's lots of them together. And so the question is, how do they know? How does a bacterium that is like a little spot here knows that it's got a lot of other little bacteria nearby of the same species? And this is what's called the process of quorum sensing in bacteria. So they'll start to do something once they have enough bacteria present. Now, the system that was first described in Vibrio fischeri, and it's also used by many gram-negative bacteria, is that this collective behavior is really controlled by the exterior concentration of an auto-inducing molecule. So in Vibrio fischeri, this is N-acylhomoserine lactone. Now, if I kind of have a look at this, so NH and this carboxylate group here. So if this O was was an OH, that would be serine, but it's homoserine. It's like got a cyclic group here, and it's N-acyl with the R function here will be different between different bacterial species. However, this is a small hydrophobic molecule that can freely diffuse across bacterial membranes. And it's produced by an enzyme which is called LuxI, and LuxI is on this operon, the Lux operon here. So this enzyme will produce acylhomoserine lactone, which will diffuse out of the bacterial cell and get outside the bacterium. Now, if you only have a few bacteria present, the concentration of AHL will be very, very low, and then nothing else will happen. You just have the basal level of expression of LuxI, and bacteria won't do anything. But if you've got a lot of bacteria of the same species nearby, they're all secreting AHL. So the concentration outside the bacterium will increase, and then it will start to diffuse back into the bacterial cell. And once it's there, it will associate with another protein called LuxR, which will induce the expression of all the genes on this operon. So you'll get more LuxI expressed, so it's going to produce even more of AHL, so the whole thing has got a positive feedback loop. Get AHL concentration starts to increase, and it induces the expression of the enzyme that produces even more AHL. So once it starts, it really starts going quickly. And one of the other proteins that are coded on this operon, on this gene here, code for the luciferase enzyme. So what is happening is here is that when there are enough bacteria present, they produce a lot of this auto-inducing molecule, and that's what turns on the expression of luciferase. And all of the bacteria are doing the same thing. So they all detect the same external concentration. So they all start the expression of luciferase at the same time. That's how they do it. So that's kind of the key to this quorum sensing mechanism. And a lot of gram-negative bacteria have similar systems based on acylhomoserine lactone molecules. So Agrobacterium tumefaciens, it can conjugate, it can form F-pili, conjugation pili, and exchange genes. Well, that's only useful if you've got some other bacteria of the same species nearby, right? If there's nobody around you, no partner with whom you might be able to share your genetic material, there's no point, okay? So that makes sense. They will only start to transfer plasmids between bacteria if there's enough of them locally. Pseudomonas aeruginosa for their biofilm formation and virulence. These are, genetic programs of virulence that start to be expressed once there are enough of the bacteria that have colonized the poor unfortunate individual has been infected. Yeah, this is a bit of a side question here, but Pseudomonas aeruginosa, does anybody know for what particular population this is a problem? People with cystic fibrosis, which is in French, what is that? Mucoviscidose. Okay, the most frequent autosomal genetic disease in the European population. So this biofilm makes it very, very difficult to eject the bacteria by coughing. Okay, it's very, very hard to get rid of them. It's particularly a problem for people with cystic fibrosis, kind of like the number one pathogen, which is a big problem for them. Okay, so that's gram negatives. Gram positives, they also have quorum sensing, but the system they use is very different. So many gram positives will secrete auto-inducing peptides, AIP, that have a similar function. So, you know, the quorum sensing by auto-inducing peptides is used by Staphylococcus aureus to start expressing virulence factors. And in Bacillus subtilis, this is what induces sporulation. So if you have a lot of bacteria that are growing densely, the nutrients are going to run out. There's no point trying to carry on growing. So they will detect that they have a lot of other bacteria nearby. Okay, time for me to produce endospores. There's too much competition nearby. Okay, so I better produce a spore right now when there's still some nutrients around for me to do this. If I wait another hour or two, that's it. It's all going to be gone. So that's the kind of behavior that is controlled by these systems. And all the bacteria will start sporulating at the same time. So auto-inducing peptides, how are they produced? Well, first there's a protein that's produced in the cytosol, which is cleaved into these small peptides, which are exported out into the medium surrounding the bacteria. So similarly, if there are enough bacteria nearby doing this, then the concentration, the extracellular concentration will go up. And these AIPs will be detected by a specific receptor on the plasma membrane, which will transmit a signal, and that will control the expression of target genes. Okay, so all the bacteria locally will all switch on the same sets of genes at the same time. Okay, so that's all I've got to say about bacterial growth in liquid culture and on surfaces. So in the last 20 minutes, what I think I'll talk about are the impact of environmental factors on bacterial growth. Now, this is kind of related to bacterial physiology, okay? So the environmental factors are what? It's like temperature, presence of oxygen, pH, salt content, water availability. And so for each of these things, you know, bacteria have got to be able to somehow survive and keep growing with varied conditions outside the bacterial cell. So in that sense, it's a kind of physiological problem for bacteria. Okay, how do I keep my proteins, my membranes functioning in different kind of environmental conditions? So this part is to do with bacterial growth. It's related to bacterial growth, but another way to look at it is that it's bacterial physiology. Okay, so how about temperature? So each particular bacterial species will have a preferred growth temperature, and it will also have a minimum growth temperature and a maximal growth temperature. So these three things are what are called the cardinal temperatures for a bacterium. So minimum, the bacterium cannot grow at a temperature any lower than that. Maximum temperature, it can't grow at any temperature higher than that. And the optimum, that's it. They love it. That's where they have their fastest growth rate. So here are some results that I found from a paper published practically 30 years ago now. So they're measuring the bacterial growth rate at different temperatures, and they can see that the optimum growth rate is somewhere between 25 to 35. Starts getting lower at about 40 degrees, and you can imagine if they tested up to 45, these bacteria wouldn't grow anymore. This is Listeria monocytogenes, which is kind of unusual because it can still grow, even slowly, down at about 4 degrees C. So that's why Listeria monocytogenes, this is the one that causes you problems when you've eaten some old ham that's been sitting in your fridge for the last week or so. Because it can, or in your, I don't know, mixture of tuna and mayonnaise kind of thing that you put on your sandwich. You prepared, done it last week, you left it in your fridge. Well, Listeria, they can still grow at 4 degrees. And they've got a pretty wide temperature range. Their optimum temperature for growth is going to be about 30 to 35, 37. So they grow pretty well in a human body. So they can infect you and cause disease. But that's really pretty rare because most bacteria that are adapted to grow at 37 degrees, they can't grow anymore at temperatures less than 10. So if you keep something in the fridge, then almost all bacterial pathogens, they don't grow anymore. Okay, so this temperature dependence of bacterial growth is kind of summarized by particular terms. So mesophiles are bacteria that have an optimal growth temperature between 20 and 40 degrees. Thermophiles, they'll have their optimal growth between 55 and 60, and hyperthermophiles, their optimal growth temperature is higher than 80 degrees. Psychotrophs, these bacteria can grow at low temperatures, but their optimal is still going to be 20 to 30 degrees. The maximum here, 35, Listeria monocytogenes can still grow up to about 40 degrees. So, this is genuinely true for most psychotrophs. Listeria can grow at kind of the high end of this scale. Psychophilic bacteria, they really prefer to grow at low temperatures with an optimum at 10 degrees, can't grow anymore at temperatures higher than 25. Some of them can still grow at temperatures under 0. So if you've got seawater, for example, might not be frozen at -1 degrees. And some bacteria can still grow. The bacteria that grow in the Arctic or the Antarctic, they will be psychophilic. Bacteria that grow in hot springs, well, they'll be adapted to the temperature of the water around it. Some of them can grow, in vents, volcanic vents under the sea at high pressure. That's why their optimal growth temperature can be higher than 100 degrees because it's under high pressure and the water can't boil at such high pressure. It's still liquid. Okay, so I've given an example of some of each type of these bacteria. This one down here in blue, that's an archaeobacteria. So many of the archaeobacteria that have been characterized are extremophiles. Okay, so how do bacteria live at higher or lower temperatures? And there are some interesting molecular adaptations that bacteria use. So psychrophiles, their proteins, their enzymes have to function at lower temperatures. And when you look at the structure of their proteins, you can see that they don't have so many hydrophobic interactions between amino acids on the inside of the protein. And they don't have so many hydrogen bonds between adjacent, I don't know, don't have so many internal hydrogen bonds either. And they don't have so much proline in their proteins. So overall, this means their proteins are less stable and they are more flexible. But all enzymes need a little bit of thermal flexibility. Otherwise, the catalysis won't actually function. So, it's important for their proteins to have a little bit of flexibility, even at low temperature. And these are the adaptations that allow that to happen. Now, on the kind of contrary, thermophiles, they've got a lot of hydrophobic residues buried inside the proteins. They've got more hydrogen bonds and they have a higher content of proline. So if you remember your biochemistry, proline is this amino acid. So there's the carboxylate group and there's the amine group. And it's the only amino acid where the amine group is in a kind of like cyclic side chain. So that means that the peptide bond between proline and the next amino acid is not so flexible. It can't rotate so much. So proteins with a lot of proline, they have a lot less molecular flexibility. And that's also what makes them more stable. So this thing about the hydrophobic interactions, you know, I hope you all remember from physical chemistry, it's the only type of chemical bond that gets stronger as temperature increases, right? Because it's entropy driven. So as temperature increases, these hydrophobic interactions that are holding the protein together, they get stronger and they can resist high temperatures. Okay, the other things that you find in these bacteria is in psychrophilic bacteria. They can sometimes express antifreeze proteins that will prevent water from crystallizing into ice inside the bacterium, which is kind of important if you're growing at -1, -2 degrees. Thermophiles will have specialized chaperone proteins that can stay stable and help refold proteins even at high temperatures. So that's all kind of like really interesting stuff from a fundamental biochemistry point of view, but it's also useful to study and isolate enzymes from these extremophile bacteria, because psychrophiles will produce enzymes that are active at low temperatures. So proteases, lipases, cellulases, and there's some interest in using these enzymes in, washing powder for your washing machine, so that if you wash at a low temperature, you can still get rid of stains. You put in some of these proteases, lipases, and they can degrade like a big stain of like fried egg that you've got on your t-shirt or something, even at like low temperature. So you don't need to use so much energy for your washing machine. For the thermophiles, they express thermostable enzymes that will still work at high temperatures. So this is useful in a lot of molecular biology applications. The Taq polymerase from Thermus aquaticus, which is a thermophile. And other enzymes that are useful are silanases, lacases, that can be expressed, by a bunch of different microorganisms. And if they are stable at high temperatures, they can be used in biological bleaching processes in the textile industry, which otherwise you would get like raw cotton fibers or something. They're kind of yellowy. To get them to be beautiful and white before dyeing them or turning them into a white shirt, there's a bleaching process. And normally this is just chemical with, I don't know, hypochlorite or something like that, and large quantities of toxic chemicals. So if you could do this by just adding some thermostable enzymes, then this industrial application wouldn't need to produce so much pollution. Okay, so there are important industrial applications of enzymes from these thermophiles and psychrophiles. So proteins are different. The other things that are different between thermophiles and psychrophiles is the composition of the fatty acids in the lipid membranes. So in order to function, a biological membrane has got to have a certain fluidity. Okay, if it's too immobile and too stable, all of the integral membrane proteins can't really move about and they won't be able to interact with each other and it won't work. If the membrane is too fluid, it's not going to be stable enough, it will fall apart. So there's an equilibrium that has to be observed to have a functioning cell. So bacteria that grow at low temperatures, they've got unsaturated fatty acids in their lipids. So the melting temperature is lower. Mesophiles, they've got a lot of palmitate. So just, C16 saturated fatty acid here. And in thermophiles, you have different kind of weird fatty acid chains like cyclohexyl fatty acids or these kind of branched fatty acids here. So membrane lipids with saturated chains and branches, and these will have a tendency to increase the melting temperature of the lipids. And in archaea, then they have a very different fatty acid structure produced with these prenyl groups, and some of them have, a kind of double-length fatty acid that's got the hydrophilic head group at both ends. So these are very, very stable and resistant to high temperatures. Now, bacteria that can grow pretty well over a wide temperature range, they've got the possibility to slightly modify the composition of their membrane lipids depending on the temperature. So I believe that Bacillus is the example. I might just better check my notes if I wrote this down, actually. I didn't write down particular bacteria, but okay, so bacteria that can grow over a wide range of temperatures, they can modify the composition of their membrane lipids. So if they're growing at lower temperatures, they will introduce, or they'll produce unsaturated fatty acids, and also modify the lipids that are already in the membrane to introduce these unsaturated bonds. And if they're growing at higher temperatures, they'll do the opposite, okay? So that gives bacteria a little bit of flexibility to grow pretty well at a wider range of temperatures. Now, that kind of thing might be important for something like an opportunistic pathogen, like Vibrio cholerae. So most of the time, Vibrio cholerae is swimming around in rivers, growing at like ambient temperature, but then it will infect a human being and it needs to grow at 37. So it's got to be able to adapt its metabolism to grow at a higher temperature. Now, this is the kind of thing that helps in terms of lipids. So we've got about 8 minutes to go. I'm just going to talk very quickly about oxygen relationships. So different bacterial species have different oxygen requirements. And you can test this pretty easily by inoculating bacteria into a long tube with a soft agar medium, which will solidify after you cool it down. And then you put the cap on, but you leave it slightly open so that oxygen can dissolve into the agar at the top of the tube. But it can't really diffuse very far down the agar here. So you get an oxygen gradient through the tube with a high oxygen concentration at the top and no oxygen down at the bottom. So what you do is you inoculate the bacteria all the way down the tube. And if bacteria only grow at the top, then they are obligate aerobes. They require oxygen for their growth. At the opposite end of the scale is if bacteria only grow down here, they are strict anaerobes. They can only grow, or they are where there is no oxygen. They cannot grow if oxygen is present. And between these two extremes, there are different kind of situations. So aerotolerant anaerobes will grow all the way up the tube, but they don't grow any better when oxygen is present. While facultative anaerobes, they can grow all the way up the tube, but they grow better when oxygen is present. So they can use oxygen when it's present, but it's not an absolute requirement. And there are also some rather annoying bacteria, which are called microaerophilic, which require oxygen. They can't grow without oxygen, but too much oxygen is toxic for them. And so some examples of bacteria in these different categories are given here. So E. coli, typical facultative anaerobe. Bacteroides, Clostridium are strict anaerobes. Campylobacter is microaerophilic, and Enterococcus is an aerotolerant anaerobe. Now, whether bacteria can survive or not in the presence of high concentrations of oxygen depends on these two enzymes, which are superoxide dismutase and catalase. So superoxide dismutase catalyzes this reaction, which will add a couple of protons to the oxygen radical, which is a molecule of dioxygen which has accumulated an extra electron. So it's O2 minus with one free electron. It's highly reactive, highly toxic. So to detoxify this molecule, it's superoxide dismutase, which will produce hydrogen peroxide, H2O2, which is much less toxic than the O2 radical, but still quite toxic. You know, you can use this to disinfect an open wound, okay, hydrogen peroxide. So if hydrogen peroxide is present at a high concentration, it's also got to be split into harmless molecules, which is the job of the catalase, which will split it into water and dioxygen gas. So aerotolerant anaerobes will express superoxide dismutase, but not catalase. Strict anaerobes don't have either one, okay? They can't tolerate any oxygen. And microaerophilic bacteria, well, they have superoxide dismutase, might have a bit of catalase. But facultative anaerobes and obligate aerobes need both of these enzymes. And the reason is because these bacteria can use oxygen in their metabolism as the terminal electron acceptor in respiration. So if it's a terminal electron acceptor, that means bacteria are pushing extra electrons onto this molecule of oxygen. And that produces this oxygen radical as a kind of harmful byproduct of aerobic respiration. So the concentrations of these oxygen radicals that obligate aerobes and facultative anaerobes are confronted with is going to be much higher. Okay, so there'll always be some hanging around in low concentrations with a high concentration of oxygen. But the high concentrations are only formed by these obligate aerobes and the facultative anaerobes when they're using oxygen for respiration. So the concentrations are much higher. So if they just had superoxide dismutase, they would accumulate a high concentration of hydrogen peroxide, which would accumulate to concentrations that are going to be toxic. So they need also catalase. Okay. So I think I would just about stop there. It's time to stop anyway. So microbe of the week. When do you want to do the next one? Should we send one out this evening or later? What do you? Huh? Tonight, 7 p.m. Okay. I'll be ready. Okay, thanks very much for listening, and I'll see you Friday, right? Yeah.